Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled New Frontiers in Respiratory Trials, AI, Lung Sounds, Remote Coaching, and Alternative Endpoints. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout this presentation by using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, and that's found on the right-hand side of your screen. If you require assistance, please contact me at any time by sending me a message using that chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available to you for future streaming on xtalks.com. At this point, I'd like to thank Clario, who developed the content for this presentation. Clario is a leading healthcare research and technology company that generates the richest clinical evidence in the industry. Across decentralized hybrid and site-based clinical trials, Clario's deep scientific expertise, global scale, and the broadest endpoint technology platform allows their partners to transform lives. Now, it's my pleasure to introduce you to your speakers for today's webinar, and we have a great audience, speakers here with us today. And first, I'd like to introduce you to Kevin McCarthy. He's the Director of Respiratory Science with Clario, and there he is. Kevin has 50 years experience in the field of lung function testing. Oh, and there's Phil Lake. He's already joined us. There we go, Phil. Hi, Phil. He's a Senior Director of Respiratory Solutions with Clario. And Phil has worked in the respiratory drug development for 25 years and has worked extensively to understand why clinical trials fail and how to gain more accurate treatment effect data. And next is Marko Topalović. He is the CEO and co-founder for ArtiQ, a company that aims to empower medical professionals with artificial intelligence to accurately and timely diagnose, treat, and monitor patients with lung diseases. And next is Dr. Adrian Marinovich, Senior Machine Learning Engineer with Strados Labs. And prior to his work as a machine learning engineer and data scientist, Adrian was an HIV epidemiologist and attending pathologist, including at St. Christopher's Hospital for Children in Philadelphia. And next is Dr. Kai Michael Beeh. He's the founder and medical director of INSAF Respiratory Research Institute. And Kai is the board certified pulmonologist and founder of the INSAF Respiratory Research Institute in Wiesbaden. Now, there he is. Thank you, Kai. Whew. Okay, I got through everyone there. I didn't miss anyone. Good. All right. Now it's my pleasure to pass the mic over to Kevin. So, Kevin, when you're ready, you may begin.
Thank you, Sonia. And I want to thank all of my esteemed co-panelists today. Here's an overview of what we're going to talk about today, and we have quite a bit to say, so let's jump in. So what I'd like to do is take a look at where we stand right now. We've all kind of weathered the worst of it, but the fallout still remains. COVID shut down a lot of hospital-based and clinic-based PFT labs that were involved in clinical trials for an extended period. And now that's over. We picked up with clinical trials again, but the recruitment rates have dropped, and so that's caused a real serious focus on how do you recruit new sites, what can we do to enhance recruitment of patients, how can we make clinical trials more attractive for patients' participation. And one of the thoughts was, okay, well, decentralize the trial. Let's do some of these measurements at home. Now, Clario has a lot of experience with home-based FEV1 and peak flow measurements, a couple of decades worth, and we can share some of that with you as well. But we know that there's a couple of things we know about spirometry. It's very effort dependent. It normally has some variability associated with the patient, some with the technique, and we'll talk more about that in a bit. But when you leave it to a patient, you say, here's a spirometer, take it home, you're liable to get more variability, and variability is sort of the bane of clinical trials. It kind of hides the signal you're looking for to tell you how a drug is working on the lungs. So other things that have happened with the reduced involvement with patients is that, trying to make it easier for them to pass their respiratory screening, spirometry. And about the only thing I can say about that is, sure, that seems like it'll work, but that increased variability that comes from relaxing standards typically drives the need for more patients. So may not be the best selection. So if we talk about spirometry, the one thing that we do know about it is it's unique amongst lung function tests. Next slide. There's the really strong interaction between the operator and the patient. If you don't get maximum cooperation from the patient, you're likely to get suboptimal results that really don't reflect the best that the patient can do at that point in time. So technique is absolutely critical. And so that's one of the things that we try to focus on in clinical trials is, get a consistent technique going. And the framework for that is the ATS, ERS spirometry standards. Those standards have been around since 1979 and they're updated periodically. They do a great job of defining, well, what's good quality? What does a good quality FEV1 look like? How do we define a good quality FVC? Now, the latest spirometry standards were published in 2019, late in 2019. And I'd like to share with you something that I learned. I was on the task force that updated those standards. And what I want to show you, looking at this right now, you're looking at a volume time tracing showing the forced vital capacity maneuver. We're starting off on the left-hand side of that volume time tracing with just tidal breathing. This is what you and I do when we're not thinking about it. And then we see the start of the deep inspiration to full inflation. This is where you really get the value of the operator involving with the, getting the patient to, it's easy for them to take in the first 50%. In fact, most of the vital capacity is easy to inhale. It's that last 5% that's tough. And the sweet spot is point number 2 there. That's the spot where the technician or the operator feels like, okay, they're full. Now I have to tell them to blow before they start leaking some of that air out. And so the patient is exhorted to blast the air out as hard and as fast as they can and keep pushing until they've demonstrated they're empty, no more volume change, or have blown for a reasonable length of time. People with airway disease often don't empty completely, but we kind of limit this to 15 seconds. And before the 2019 spirometry standards, this was the end of the spirometry maneuver. And as the 2019 guidelines, one of the things that we did, myself and several others of the task force, felt that there was a consistent and turns out quite common spirometry error, starting the forced exhalation before the patient really inhaled to full inflation. And it turns out that's a great way to get a good start of test. If you go from, but if you might not get them up to full inflation by doing it. So phase 4 here is this rapid inspiration back to full inflation. And I can share with you that I've been doing that since the late 1970s. That's the standard way of doing things at Cleveland Clinic. And basically, this is something that's pretty easy for almost every patient to do. In fact, they really want to breathe back in again at the end of the forced exhalation. And it just requires a little bit of coaching to get them to breathe it in quickly and to keep breathing in until they're absolutely full. So why do we care about this forced inspiratory vital capacity? Well, it's really important. It's the first time in spirometry that we've had an objective means to really verify that forced exhalation did start from full inflation. And without spending too much time on it, you're basically comparing what they just breathed in to what they just exhaled. You can see on the graph, they share common points of measurement. They both have the common point of the end of the forced exhalation, and the other is full inflation. So simply put, if that FIVC is much larger than the FVC, even if that FVC looked by all other criteria to be perfect, then you don't want it in your clinical trial. That is, it could have a great start of test, great end of test, and it might demonstrate repeatability. But if it didn't start from full inflation, it's not representative. It's not going to tell you what the drug is doing at that time. Now, that's not the only reason why you might have variability. You might not have the right patient in that file. That happens more than you'd think. You don't have the patient's name there, so it's pretty easy to you know, to take that study ID and have another patient in the seat and not realize it until the measurement is over. Medication problems, either a problem with the washout or a problem with the inhalation technique. We try to make sure that a consistent time of day is used for testing. And the last point here, consistency of technique, that's really important. Now, one final thing I want to share with you on this next slide is something I never anticipated. I thought, hey, this phase 4 is really going to clean up clinical trials. The variability will reduce. And many sites, when they embrace it, it does exactly that. We see very consistent results from one visit to the next. And when there is a change, we're pretty certain that it really is, it's the drug that we're looking at, or the lack of drug. What you're looking at here is 8 flow volume curves from the single measurement. Those flow volume, or the FVCs from these 8 measurements or 8 efforts, vary by 0.8 of a liter. And yet the FIVC shown below the horizontal line, the volume there, would suggest that they all started from full inflation. What's happened here is that the operator coached the patient to breathe in slowly so they could easily, when that inspiratory flow volume curve matched the point where the forced exhalation started, they could hit the button to stop the measurement. And what happens then is good, you grade that data as good because the FIVC matches the FVC that's being reported, when in fact it's bad data. And so that's what can happen in a single measurement. Now I'd like to get Phil to share his expertise and tell us what happens when things like this are occurring in a clinical trial.
Thanks, Kevin. Let me just advance the slide. Yes, thanks for that introduction. What I'm going to do now is try and just kind of change the theme a bit and start thinking about home spirometry data and really real world data. Now it's fairly easy to take a little sensor, flow sensor, and Bluetooth it into a smartphone and basically start recording lung function data. And it's relatively easy again if you take a small site, one site, small number of patients who are used to doing lung function data, you can get accurate data between the site and the sensor. And in small numbers, that's not a great problem. The problem is that you can't run clinical trials like that. So in a clinical trial, you're generally dealing with hundreds, if not thousands of patients, and you're dealing with many, many sites, so tens into the hundreds of sites. So you get a lot of variability in terms of the individual patients, because there's a lot of them, and even how those patients are actually trained at the beginning, because you've got a lot of different technicians actually training those patients up. So what I wanted to do is try and show you what real world data looks like. And so there's not many trials that have run like this, but we have a nice example with the GSK CAPTAIN study. So this is all published data. Now, what they did in the CAPTAIN study is it's a standard clinic-based study. So what you can see in section A here is the results from that. So they were taking a dual combination and a triple combinations therapy and actually kind of testing that over a 6-month period. So you can see the individual doses here having incremental increases. So low dose dual into low dose and high dose triple. So it's exactly what you'd like to see. So good sturdy data. What they also did though, is they did this kind of retrospective review, rerunning the trial, if you like, using one of these home sensors. So what they did, instead of having the clinic visit, they actually took the 14 days of home data generated by the patient on themselves with the self-coached, and took the average of that for their baseline. And then at each subsequent visit, they then took 4 weeks worth of data from the patient's own home setting, took the average of that. And so you generate this composite of effect as you go through the study. Now you can imagine this might kind of dull the sudden increase at the beginning of the study because you're dealing with average data. So it smooths it out a bit. But even considering that, if we look at the overall treatment effect on this study, the thing that immediately jumps out is, yes, you can see the same kind of progression in terms of dose effect, but you get a lot of variability here, and the doses appear to be much closer together. So you don't seem to get the granularity of effect that you get when you're dealing with the clinic data. And you'll notice with the confidence intervals, a lot of these are overlapping, so that the statistical significance is reduced as well. So the reason behind that, I mean, it's partly the way that the data is collected, but it's also the variability. So if you look at a blind outcome plot of that data. So what this is the difference between the clinic and the home data. So you take the clinic FEV1, subtract the home FEV1, and you get a value here, so either minus or positive. And what you do is plot that against the average of the two. So effectively what you're seeing is the variability between home and clinic for different lung sizes. So from small, more severe patients in general up to kind of larger patients. And what we see here is that the average variability between the two is fairly close to zero. So it's a little bit of a bias in favor of the clinic, which is what you'd expect because you're getting coaching in the clinic, and in home, the patient's doing their own coaching effectively. So they're just trying their best. The thing to notice here, though, is that the outliers in this kind of in this plot, some of these outliers are absolutely huge. So they're actually, larger in clinic and larger at home. So it's not just one way. But if you're looking at the magnitude of these, some of these are 3.5 litres. And when we consider the clinic treatment effect as 150 millilitres, you're looking at some of these outliers being 20 times the treatment response of a normal patient. So when you've got small groups of patients, if you're trying to differentiate between subgroups, this kind of variability is really a killer, because this can actually start, if this data isn't evenly distributed, it starts to completely modify your results. And this is the problem that we really try and face in a clinical trial setting. So let me take you down a step and show you a bit more detail. So what you can see here now is an individual patient. So I've got two patients, and this is the kind of patient that are typically enrolled in these kind of studies. So it's an asthma trial. Now in this trial, what was happening is the patient was in clinic to start off with over a period, and they were generating lung function every day. So each of these vertical lines is in this case is FEV1. So this represents one session worth of testing. And you can see their lowest value in that session is 1.7 litres, and the highest is 3.7 litres. And what this represents is that the patient comes in, does their best, that the coach is saying, well, look, you need to do better than that. You need to breathe further and blast out harder. And eventually they're pushing them up and up and up to get their personal best at each session, if you like that analogy. So technically we end up with data around about 3.7 litres. The patient then goes home. Now, if you consider this, they've actually had training through all of this period on how to do good spirometry. They go home and they're left to their own devices. So the medical device will actually feed back, a few elements of acceptability, so they'll know what to do. But if you look at the difference in the data, it's massive. So the variability in each session is much reduced. Now, if we just looked at this in isolation, without looking at this data, we'd think, well, okay, this is pretty consistent data. This is probably fairly accurate. The reality is it's nowhere near accurate. I mean, it's at 2 litres, and we should be at 3.7 litres. You can see a few other kind of variables as well, and I'll talk about those in a minute. If we go on to look at another patient, now I should have actually put one in for FEV1 as the same, because the value is actually you get the same kind of effect. But what we see here in clinic is that the patient's variability is much reduced, and that's very consistent during the day. Now, when they actually go home, again, there's occasional perturbations, but in general, they carry on with roughly the same lung function and the same level of variability. So there is actually a drug effect here. So we are expecting them to potentially improve slightly if they're on active medication. So this is one of the core problems we face. So it's really understanding, well, how do we actually deal with this kind of sub-maximal data? So there's two things that we can do really. One is if we feed back to the patient on a more regular basis and potentially bring in some kind of learning effect. So what potentially will happen is that they'll get better and they'll move from this level closer to this level, and they may even achieve it. The trouble you have there is you have this rising effect from this point onwards. Now, if this is your baseline and they're gradually getting better over time, what you're seeing is what appears to be a really good treatment effect. The reality is this is just a training effect. Now, how do you differentiate between an increase in FEV1? You need to know whether it's a treatment effect or if you need to know if it's a drug effect. And what we're trying to work out is the drug effect. So this kind of ongoing learning is really a disaster for trials. You really need to have a stable baseline, which is reliable and accurate, which allows you then to see any improvement relates to the drug and not other elements of variability. So in a home-based setting, there's a couple of other things as well. So one, we can't control as much as we can in the clinic. So patient may well be taking their rescue medication at home before they've done their test. So there's all of that kind of material variability is also there. What we've also seen as well in home setting, particularly in adolescent studies, is that the patient doesn't always want to do the test. And we've actually seen instances where the parent steps in and does a test for them. So we've actually worked this out from, calling up and finding out afterwards, but you suddenly get this massive improvement. Equally, you can have a massive drop, and I'll show you what drives that. It's really largely technique that's the driver of it. So the last point I wanted to make, which is actually another hurdle for us, is the problem of patient burden. So, we can feedback and get them to do more and more tests, but the thing is, it's actually quite hard work doing these tests, and I'll demonstrate in a minute. But the last thing we want to do is to make it so hard for the patient that they really drop out of the trial. So what we need is a balance between optimum data and variability and patient burden. So let me move on to the next trial. The next slide. So what I've done now is gone back even further. So we've talked about the trial, the patient, and now we're talking about the individual effort. Now, don't worry if you don't understand what these are, but I'll explain them. So this is a flow volume loop. This is traditionally the way we looked at data. So if they all sat on top of each other like this, it was generally considered to be consistent data, and consistency used to be considered to be accuracy. So they must have been breathing in at the right level every time. When you look at that data in more detail, if you look at this is the same effort, look at the full volume flow chart, including the bit before they start to blow out, so the bit over here, we see their tidal breathing, which is going up and down, which is what you and I are doing now. Now, in a good test, this is the same patient at the next visit, you see their nice tidal breathing, and what they've done is they breathe all the way in, right the way up to the top, and then they blast all the way out again. And this gives you, maximum inhalation. What they've done here is, from you and I both now are sitting here with our tidal breathing, what they've done is they've gone, they've blasted the air out from their tidal breath. And so what you get is a relatively short expiration. Now, as long as there's no cough or hesitation and they breathe out until there's a plateau, there's no more air, that's considered good, or it used to be. So this is why Kevin was really keen to actually check to make sure they'd breathe all the way in. Now, if I do it properly now, I'd go right up to the top and now I'm going to stop the crop early because I could keep squeezing out for another 5-10 seconds. But you can tell that the effort of doing that is actually quite hard, but technically the amount of air I'm breathing out is massive. And this is the problem we face. So not everyone thinks that this is full. They're breathing halfway and they think, well, that's pretty good, I'll blow from there. That's what causes the variability, and that's what really starts to undermine trials. So let me show you a solution I think we have for part of this. So what we've done is we've actually taken the best bits of the clinic and we've really transported them into the home. So what we do is we still give the patient a little flow sensor, a provisioned phone for them to take home with them. And what we then do is we get the site via the Clario portal to basically initiate a kind of telemedicine visit with them. So the site will then start the event, they'll see the patient on the screen, they can do their normal chat and preamble, but then when they move into the testing mode, what they then see, it's a little bit like a head-up display for a fighter pilot. They see their standard spirometry system, and then they see the patient who they can talk to and hear. And the most useful fact here is that because of the latency between the two, that you can actually see exactly what's happening in terms of the flow loop as the patient's actually doing it themselves. So this is good enough for you to actually be able to coach the patient, both from looking at them in this and actually seeing the flow volume loop. So you can see that they're at plateau. You can see that they're just getting the last few bits, and then you can push them into exhalation. So let me go on. So effectively, we've run about 4 studies like this now with these kind of patients of different sizes. Now, I can't share with you the data because it's still being analysed and it will be published, but I can actually give you a kind of top line summary of them. So technically what we see is the data is virtually equivalent between site and home when you're actually measuring it in clinic and you're measuring it via the remote coach session. The thing is, you do see some variability, but what generally happens is if the patient's variable at home, the patient's also variable in site. And so this probably won't be a surprise to you. So technicians who manage to generate really good, high quality, reproducible data at site manage to do it exactly the same with this technology. So it's no barrier for them to actually achieve good quality. The ones that actually don't do so well at site, this technology doesn't make them superhuman and improve their abilities. They're still bad when it comes to doing it at home. So it's really down to the quality of the technician and the training that will actually drive the quality of the data. In terms of the patient experience, the feedback is good. I mean, most patients liked having reduced burden. Some actually like going to site, which is we can't get around that. But it definitely reduces the time and makes it more efficient. And the other thing in terms of data plausibility, which is the real positive, is that you have your own personal coach there. So every data point benefits from having that optimal data and someone to actually make sure that you are pushing yourself as hard as you can be and can actually check between different visits to make sure it's happening. So the learning effect is massively diminished, if not absent. So hopefully you found that interesting. I'm going to kind of move forwards now and hand you over to Marko, who's going to take you through some of the new interesting areas for AI. So Marko, over to you.
Thank you, Phil. Thank you also, Kevin, as well, for a very nice introduction. So happy to be here and share the offering and share you the results of the studies and the research and the products that ArtiQ is putting on the market. So it's good to basically remind ourselves to recap some things that also Phil and Kevin already said, that why is actually spirometry important in the context of clinical trials? Well, because FE1 and FEC are the mandatory endpoints in the respiratory clinical trials, because spirometry is also criteria for inclusions often and exclusions, and also because spirometry helps us to evaluate the effectiveness of the drugs. So all these three elements are actually highlighting why the quality of this spirometry is actually pretty important, or utmost importance, to be achieved in the clinical trials. And that's why to achieve that, to achieve the highest quality of the data and prevent certain problems to happen in the future, we are doing certain overread. So the overread is important to because spirometry, as also Kevin and Phil said, it's very patient dependent. You need to blow out the maximum. So you need a good patient effort, and you need a good coaching as well. If you don't have that, you will have faulty data, you will have increased data variability in the studies, and that's going to have another effect. You will have to have increased number of recruited patients, additional costs, and eventually a risking of trial success. So ATS and ERS gave us certain guidelines how to secure the highest quality of data. And interestingly, half of these rules in these guidelines are numerical. So it's something that we can make a very simple analysis whether it's correct or not. But half of these rules are also quite subjective or visual. So it is left to very qualified and experienced technicians to be able to judge the quality. Was it performed well or not? And if you don't do that, we're going to have really high variability between different subjects, different technicians who are judging whether this is a good or bad quality of spirometry. So how can we move that? So how can we improve that process today? For that, we need to understand how the process looks today. So we have a patient who is at home doing spirometry or in the clinic, and today we're going to have the expert who's going to analyze those numeric and visual rules, and in the time of up to 48 hours, the expert is going to provide to the investigator with evaluation, overread evaluation, whether the spirometry was performed satisfactory or not. And the process has a certain pain points because we have that long turnaround time. That could be a problem sometimes not. We're going to have also this possibility to have human errors and variability between the different experts, and that cause eventually has some side effects. We're going to have a patient revisit the clinic. If it's in clinical trial, we're going to have maybe some late decisions for recruitment and randomization, and we are risking data contamination in the study. So what we hypothesize as a company, we hypothesize and then build a product, is that artificial intelligence can learn that the same, can learn this in the same way to look as the experts into this visual aspect of the quality control, and then provide the same kind of consistent reply. And it's interesting because we actually expose artificial intelligence to the number of spirometries that usually the technicians are not seeing in their lifetime. And this is exactly then the solution that we are offering today. It's artificial intelligence based overreading that is instant, so it's automatic and instant. Be that we are enabling that innovation in hybrid clinical trials, so be it in clinic and at home. And we believe that also the huge values of this is pairing that with the coaching system of Clario at home trials. By giving these quick and consistent results, we are also improving the patient experience, because the patient will have to reduce revisits. We're going to reduce actually the number of revisits, and overall the side effect or the effect is we're going to have more efficient clinical trials. So how this improved process look today? So the spirometry is going to be performed either at home or in clinic. And then the AI will in 3 seconds look into this both numerical and visual data points and provide immediately the results to the investigator. And these are just some couple of examples that I prepared here for you. Is for spirometry, and then where we highlight in red as educational component as well what AI thinks went wrong. And I think it's an interesting example, because Beeh criteria, which is a proxy for hesitation or a wrong start during the spirometry maneuver, was actually green. So that means it was satisfactory. But AI still identified that there was a certain hesitation that was happening, and that the spirometry should just be repeated. Or this is also a very interesting example because there is no real proxy of whether there was a sub-maximal effort or a variable effort in spirometry. And we identified here that actually AI was able to detect that the patient did not give the maximal blast that was expected. And then highlight as well and show back to the either patient or to the investigator. So probably you also wonder, can AI detect something else in this curve? So we have the step 1, which is the improvement of the traditional overreading on that patient level. But also, as Phil said, in the clinical trials, we have sometimes hundreds of sites, depending on the disease that we're looking for. So what AI is able to do that in real time is also look serve as a risk-based quality monitoring of the complete site. So not only patient level, but also site level. And in that way, we could warn about consistent errors. We could warn about whether the site was not trained enough, or then go back to be retrained, or can also detect certain fraud that might have happened in that site. And finally, we can also serve in non-respiratory trials as a monitoring of the patient safety and the side effects, as it can look into the fingerprints, whether some disease left some fingerprint of it on a spirometry curve as well. We also did quite some studies to show to further validate this technology. So something that we are pretty proud of is the academic study published in the ERJ, which we did together with the GLI to revise the predictive equations in the Chinese population. Then we also did some studies in the clinical trials where we show that actually AI has that high accuracy and consistency in the COPD and asthma trials in this case. And also, as a very important topic of today, home-based spirometry, we also show, we just presented at the ATS that in the arm that was used ArtiQ as a overreading was having a significantly improved quality of spirometry comparing to the arm that didn't use the AI. So to conclude, we believe that that AI is the new normal of spirometry overreading and the quality control, and it's not a promise of the future. It's actually something that is happening already today. So it has been used or is being used in 11 different studies, be it from phase 1 all the way to the phase 4. we believe that that's going to be that missing enabler of the home spirometry and actually monitoring of quality beyond the individual. And finally, we are working, and importantly, because that's the topic of this whole webinar, is that working closely with Clario. We are working closely with Clario to bring that AI-based overreading to the clinical trials already today. So with this, I would like to thank you. You can always reach out for the further questions, but with this, I would like to thank you and give the next presenter, Adrian, Dr. Adrian Marinovich.
Turn on your mic.
Thank you for reminding me. Thank you, Marco, and thank you everyone for having me here. So I'm going to talk a little bit about how and why we see remote lung sound monitoring fitting into clinical trials. So for starters, what would make lung sounds new or novel as biomarkers and endpoints? As measurements, there certainly are. They certainly are can be considered biomarkers, but auscultation as part of a physical exam is definitely not novel. So what I'm talking about here specifically is the remote longitudinal capture of lung sound recordings by a wearable sensor, and how events systematically detected in those recordings can be considered novel biomarkers. By lung sounds, I'm pulling together coughs as well as the more properly intrinsic lung sounds of wheeze, rhonchi, and crackles. But of course, there can be other sounds captured as well using this approach, including non-lung sounds. You can capture heart sounds, stridor, and so forth. But with lung sounds captured in this way, we can provide objective measures alongside subjective measures. For instance, we can provide cough counting alongside patient self-report of their coughing. We can also complement other existing objective measures such as spirometry, and that can help to strengthen evidence gathered in a clinical trial, and as well as a potential to gain new pathophysiological insights into disease, and as well into treatment response for that as well. So at Strados, we make a wearable biosensor. It has a membrane on the body-facing surface that is similar to what would be found in a stethoscope, and it provides that continuous remote capture of lung sounds. It's substantially equivalent to what would be obtained from an electronic stethoscope that would be used in-person episodic exams by a healthcare provider. The battery life is long enough to allow that 24-hour capture, up to 24-hour capture of those sounds, as well as it's passive. So once the patient has the device on them, they don't have to take any other actions aside from removing the device at the end of a recording session. So this all allows for us to do detection of cough events and other lung sound events. And currently now we are working with 2 companies in providing cough monitoring for clinical trials. So the biosensor is held in place with an adhesive tegaderm to the patient's chest. They can then go about their day while the recording progresses. The recordings are transferred securely to the cloud using a paired smartphone. And then once up there in the cloud, then trained labelers using semi-automated processes can perform the event labeling, and those then can be viewed, such as cough counts, in an online web portal. So that's the overview of the technology. It has been used, we have used it in a range of patients and volunteers. It's been very well tolerated and used in both inpatient and outpatient settings, both with patients with COPD, asthma, heart failure, as well as respiratory infections. And we've got over 47,000 sounds and counting detected and validated by clinicians. But while this is all all great to be capturing the sounds and a lot of focus on the sound and those sound events that are labeled up. What's I think most interesting is what's the connection between those and the clinical course of the patient. So in this case study, there was an adult male. He was discharged following treatment for asthma exacerbation in the hospital, went home with a device. He was wearing a device at home in an ambulatory setting. And he was also tracking his symptoms as shown here, his dyspnea, his shortness of breath. And what we find is that the wheezes and rhonchi events that are being detected are tracking along with an increase in his shortness of breath towards the end of this interval. And what's also interesting is at the end, a day after recording ended, he was readmitted at the time previously when there was a peak in those wheeze and rhonchi events. In another case study, the patient was wearing the device in the hospital. He had been admitted for decompensated congestive heart failure. And you can see here, we're tracking the crackles, and he's at a baseline during most of this admission. Just towards the end of the admission, actually, the crackle events were shown to be increasing. I should note that as part of the protocol, these data were not being shared actively with the clinical team. He was discharged home. He did return for second admission, during which his condition deteriorated, and we do see concomitant with that peaks in his crackles events that we're detecting. These are individual case studies. To take things from another angle, we have here patients presenting to the emergency department. They have respiratory distress, most of them with asthma, also some with COPD. And after treatment, the peak expiratory flow shows to the right of this vertical line a favorable response to that treatment. But what's interesting for us is that in the counts of events, the cough and the wheeze and the way they change post-treatment, actually, we're seeing some on the red line and above indicating actually they are not showing decreases in those sound events, those wheeze and cough sound events that would correlate with the favorable PEF result, the peak expiratory flow result. So this indicates potential that the sounds are providing additional information that is not being provided by the peak expiratory flow. In another study, we collaborated with researchers who were using a smartphone-based app to collect patient self-reports in an ambulatory setting as they were at home for a variety of symptoms, including cough. The patients 16 patients wore the REST biosensor, and were also providing we're also reporting reports of cough. And what we found is 3/4 of those patients, the self-reported cough data did not match the validated biosensor cough detection, demonstrating that there's value to this objective evidence provided by this kind of biosensor in outpatients where the self-report data can be unreliable. So far, I've talked about acoustic events, sound events, but also worth noting that the biosensor does also have a motion sensor. And while the algorithms that can be applied to REST biosensor data are not themselves FDA cleared, the integration of acoustic measures with motion-enabled measures, such as respiratory rate and sleep-wake cycles, can provide a richer set of biomarkers to explore new endpoints in clinical trials. So to wrap up, the acoustic and motion information as well, provided through longitudinal remote recordings of this kind, can add a new dimension to the evidence captured in a trial, and especially in relation to treatment response. Once further validated, these biomarkers may provide the basis for development of new surrogate clinical endpoints as well. And finally, in the specific use case of cough monitoring, we found that they add value as objective measures opposed to self-report. Thanks very much, and feel free to follow up with us. Next will be Dr. Beeh to continue on with alternative endpoints.
Thank you very much, Adrian, for this very fascinating talk. Now my final talk is going to basically conclude this session, and I will go back to the beginning, if you like. So given the talk of Kevin and Phil and the limitations of FEV1 enforced spirometry, not only technical limitations, but also the limitations of spirometry to fully reflect lung pathology, you may ask yourself, Well, isn't there anything else there that can be an alternative endpoint for just force spirometry? Can we not measure other things than just FEV1 and FVC? And the good answer to that is yes, there are different things out there. And I'd like to touch on 3 things which I think are very important, not only in clinical practice, where I use these parameters already on an everyday basis, but as a clinical researcher, I can also tell you that these parameters are increasingly used in clinical trials, both as outcome markers, but also as stratification tools. And that's the basic point of my talk. So we will touch on 3 things: oscillometry, DLCO, diffusion capacity, and FeNO. So oscillometry is touching the same aspect of lung pathology as force spirometry. It deals with airflow limitation, whereas diffusion capacity goes beyond that and looks at another very important aspect of lung health, which is the ability to absorb oxygen. And then finally, we come to FeNO, which is an inflammatory marker indicative of certain aspects of inflammation going on, particularly in allergic or asthmatic airway disease. So let's start with oscillometry. So I grew up in an environment where we used to measure airway resistance for obstructive airway diseases, always with body plethysmography, which is a nice technique, but it is expensive and the technique is difficult to standardize and to provide, for example, in a setting of a clinical trial. And this is where oscillometry comes in. It is a small, relatively cost-effective technique to measure airway resistance in patients with or without underlying airway diseases. And in contrast to force spirometry, it is more or less totally effort independent, and it is there's very little burden associated with it to the patient. So that means that we could consider this as not only an alternative to spirometry, but as it measures other parameters, you could also use it as an, if you like, complementary outcome parameter to conventional spirometry. So this is also something that could be suitable in small children, where you all know that we cannot obtain reliable forced spirometry data. And quite importantly, this machine does not measure only the presence, yes or no, of airflow obstruction, but it can also give you some information about the locus of airway inflammation. So where does the airway dysfunction come from? Small or large airways. The technicalities about that are complex. However, more importantly for the operator, it is a very easy operational process. Basically, everything you have to do is tidal breathing for whatever 10, 15, maybe sometimes 30 seconds. So very little burden. Now, what the machine does is that it sends or superimposes sound waves over quiet breathing in patients. And that oscillation will then blend with the tidal breathing curve to give you this breathing pattern. And the machine can generate sound waves at different frequencies. And this is where it becomes very interesting, because we know that sounds at different frequencies travel forth and back the airways to different regions. So that, for example, high frequency sound waves travel to central airways, whereas low frequency sound waves travel to peripheral airways. And that reflection will then give you information about the locus of pathology, small versus large airways. So this is how a typical pattern looks. You see differences in resistance, and the second parameter that we measure is reactance at different frequencies. So you see this typical slope, and you can derive the important data from this slope. The first thing, as I told you, which is very important, which we measure, is resistance. That's a marker of airway caliber. And the second thing you can measure with these machines is reactance. Reactance is, very roughly speaking, an indicator of elastic properties in the lung. So it would change when the lung either becomes stiff or the lung becomes less elastic, for example, like in patients that have emphysema. So I give you one example here, just to give you an idea how a typical pattern of a normal subject and somebody with airflow disease will look like. So this would be a normal pattern. You can see that the resistance slope is flat, so there is no increased resistance, and the resistance is identical at low and high frequencies. So the slope is 0. And then on the other hand, you see reactance. You see that reactance increases with frequency, which is a normal pattern for somebody without lung disease. Now let's look at a pattern of somebody with peripheral airway obstruction. Now you will notice two things. First, the yellow line for resistance is elevated. So it's above normal for all frequencies, but it is more pronounced in the low frequency range. So R5 has a larger increase in resistance than, for example, R20. And the slope between R5 and R20 is also elevated. It's not zero. So that means you have a dominant obstruction in the peripheral airways, whereas it's less predominant in the large airways. On the other hand, you see that there is a uniform decrease in reactance throughout all frequency, indicative of loss of elastic properties of the airways and lungs. Now, how do I use this in clinical practice, and how do I believe that this could be very useful also in clinical trials? You all know COPD. COPD is not one disease. COPD is an umbrella term. There are patients within the group of COPD patients that have more airway predominant or bronchitic disease. And on the other hand, you have patients that have more predominant emphysema, so loss of elastic properties of the lung. Now, this is an example of two patients. One is called Lady Airway Narrowing and one is called Mr. Emphysema. Now, if you just look at conventional spirometry, you will find that they are identical. Both of them have 38% of predicted value for FEV1. You would say that's COPD at the same level of severity. However, if we then use oscillation technique as an adjunct, you will find marked differences, telling you something about underlying pathology. Now, Lady Airway Narrowing has a marked increased resistance, particularly at low frequency. So clearly predominant small airway disease, whereas reactance is more or less normal or only slightly decreased. On the other hand, you've got Mr. Emphysema, who's got normal resistance. So there is no issue with airflow obstruction in this patient. All you see is that there's massive decrease in reactance. So that means, in other words, that FEV1 reduction in Mr. Emphysema is the result of elastic recoil, whereas in Lady Airway Narrowing, it is really a problem of airway resistance. And it is very unlikely that both patients here will behave to a treatment in the same fashion, given that they are totally different mechanisms of airflow obstruction. Now, if we want to measure that in a clinical trial, it's always important to know what is a change which is relevant, and that is good news. There's been a recent publication where for the first time, minimal clinically important differences have been established for typical parameters of oscillometry, resistance, and reactance. You can read this up. So this has been anchored to clinical relevant changes in other parameters like asthma control or symptom scales. So this is now for the first time really reliable data to see, okay, is a change in resistance or reactance of this and that is something that is not only measurable, but is also clinically meaningful. The second point I'd like to touch on is the diffusion capacity. Now that's very important because it reflects the ability of the lungs basically to absorb oxygen from the air that you're breathing in and out. That's a very fundamental part of lung health. And it's quite often disturbed in many diseases. So for example, in interstitial or alveolar lung diseases like lung fibrosis or interstitial lung diseases, it is markedly reduced. But it can also be reduced in COPD, where you have the presence of emphysema because of the destruction of alveoli. And there are other diseases where this is also reduced. It's a very simple and it's a non-invasive technique. It only takes a few seconds to give you an idea about this important physiological parameter, for example, to study parenchymal lung abnormalities. Again, two examples where it is already an established marker, for example, in IPF, interstitial lung diseases, it is a prognostic marker. The worse your diffusion capacity, the worse is your prognosis. It is also an outcome parameter. It changes with treatment. And on the other hand, on the right side, you see that it is a very good, very sensitive marker of lung damage, for example, in patients treated with cytotoxic drugs. So this could also be a safety parameter. In this study, patients reacted with a decrease in DLCO before they had a pattern in imaging or they had a reduction in lung volume. So also a very sensitive safety parameter for drugs that could affect lung health. And then finally in COPD, as I said, it reflects emphysema, and it may be possible that in future clinical trials we stratify patients according to preserved or lost diffusion capacity. So patients with more or less emphysema, because it has been shown, like in this example, that if you, for example, treat patients with inhaled corticosteroid, those who have preserved TLCO, so less emphysema, respond better to the drug than those who have fewer DLCO, so have more pronounced emphysema. Differential treatment effect. Well, finally, exhaled nitric oxide, FeNO, fractional exhaled nitric oxide, an established non-invasive measurement to study inflammation in the airways. Nitric oxide is actually produced in the airways, particularly in states with allergy and asthma. We can now also use it to diagnose asthma, at least as a supportive, but very importantly, it also is something which is responsive to treatment. So we can study not only the inflammation, but the changes of inflammation after we put patients, for example, on a trial in a treatment, and a treatment in a trial. So this gas, nitric oxide, is produced in asthmatic airways. For example, if you have increased cytokines powering inflammation, then you will get an expression of an enzyme, which is called iNOS, the inducible nitric oxide synthase, and that will produce nitric oxide. For example, interleukin 13 is one of the interleukins that promotes iNOS expression. If you treat patients, this expression goes down again. So how does this work in clinical trials? Well, on the left side, you see how the fraction of exhaled nitric oxide is responsive to inhaled corticosteroid treatment. So the higher the dose, the lower the FeNO. But it can also be used in conjunction with non-ICS drugs as a basic marker of inflammation in asthma trials. On the right side, you see a study with a non-ICS drug, and you can see that there's a nice dose response reduction in FeNO according to the dose of the investigational drug. So that is something which complements the measurement of function very nicely, particularly in airway diseases like asthma and allergy. So at that stage, I'd like to hand over back to Sonja.
Okay, well, thank you very much, Kai, Kevin, Phil, Marko, and Adrian for that very detailed and insightful presentation. I hope everyone enjoyed it. Now, I know we're running a little bit short on time, but we're going to try to squeeze in a few questions. So if I get the other speakers to put their cameras on now, and I will jump to these questions. And as a reminder to our audience members, please do send your questions through. I know we don't have time to answer all of them, but the team at Clario will follow up with you after this. So I did receive a bunch of questions while you were speaking. So let's start with this first one here. This looks like it's for you, Kevin. This audience member is asking, how can a spirometry test have repeatable FVCs and FEV1s if it didn't start from full inflation? I've been doing spirometry for 16 years and was taught that repeatability was not possible unless the patient was at TLC. What do you say to that?
I say that I'm so glad they brought that up. I also was taught the same thing, that you would get repeatability only of, how could you get repeatability if at all these different varying levels of lung inflation? So now we know, now that we have an objective marker that tells us whether or not the patient truly is at full inflation, what we found out is all that's required to get repeatable results is consistency. That is a consistency in the delivery of the instructions and the patient's response to it. So you don't even have to, if, for example, you're not looking at the patient for feedback, you're just saying, okay, taking a deep breath and blow, and use the same word, same cadence, you're likely to get repeatable results. Thank you for the question.
Thank you, Kevin. Let's jump to this one. Looks like it's from Marko. Is the ArtiQ solution already available in the Clario platform, and can we already apply this in our next trial? Marko.
Oops.
Mike, yeah.
Yeah, sorry for that. So I'm also glad for that question. So that's a great question. So the answer is very simple, yes.
Okay, we're getting right through them. All right, let's go to the next one. This is for Adrian. Can you distinguish the patient's cough from the cough of others nearby?
Thanks. Yes, so there are characteristics in the biosensor data that enable us to differentiate between an internal cough, cough of the patient wearing the device versus coughs of people nearby.
Okay, thank you, Adrian. And then Kai, can FeNO also be used to support a diagnosis of asthma in a trial, or is it just a response marker?
Well, as I said, it's both. And now for the first time in a German guideline, it's mentioned also as a supportive tool for diagnosing asthma. Should not be used alone, but for example, if you want to really make sure that in an asthma trial, those who are included do have asthma. You could also use, for example, a certain signal or pheno as an inclusion criterion.
Okay, all right. Thank you very much, Kai. Adrian, back to you again. Can you tell if the patient is not wearing the device?
Yes, for example, if the patient's even quite still sleeping, for instance, we can distinguish between that. And if the device, for example, is just sitting on the bedside table, we're able to differentiate between those two things.
Okay, great. Thank you. Okay, Phil, back to you. What is the biggest challenge we face preventing us from gaining more accurate lung function data?
Oh,
Wow.
Right.
in 10 seconds, yeah. The biggest challenge, I guess it's probably education in a way. We need to actually have operators or technicians really understand all the drivers of variability. So variability is the thing that's stopping us doing it. There's all sorts of things that drive it, but if the operators really understood how to exclude all those variables, you'd have accurate data. But I mean, the key thing then is that you may not need to strive for this. You may not need to go for perfect data. It's always a compromise, but that's a talk for a subject for another webinar.
Yes. Okay, Phil, don't go anywhere. I'm going to squeeze one last question in here. This person in our audience is asking, are decentralized trials the future for respiratory drug development? Your answer, please.
Partially, yes. I think there's a lot more we need to understand about it, but I see a future where you basically use decentralized trials for certain areas where the variability is less important. But I mean, if you want to really understand like dose effect in dose ranging trials, you're probably better off doing a central clinic version or a remote coach version, because then you can really control variability and see what the drug is doing. But you're just looking for average effect and you're looking for an exacerbation signal, decentralized trials are probably much more suited to that.
Okay, thank you. All right, so I think we squeezed as much as we could in, but remember everyone, we will address your questions after this presentation, so Clario will follow up with you. I just want to say thank you very much to those people who sent in the questions as we've reached the end of the Q&A portion of the webinar. If you have any further questions, please direct them to the email addresses that you see here on your screen. Okay, thank you everyone for participating in today's webinar. You will be receiving a follow-up email from X-Talks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. Now give me a few seconds, don't go anywhere. I'm about to send you a link in your chat box and you'll be able to view the recording of this link, this recording with that link, and also share this link with your colleagues once they register for the recording as well. And get even more out of today's presentation because we do have supporting materials that are available under the handouts tab, and we have 3 there for you. So please go ahead and check that out. So I encourage you to do that. Now, please join me again in thanking our speakers, Kevin, Phil, Kai, Marko, and Adrian for that insightful presentation and for answering all your questions. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at X-Talks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now. Bye, everyone. Thank you. Bye, Kai, Kevin, Phil, Mark, Adrian. Bye, everyone.
Thank you, everybody.